Rosier identified the sample plant as a fern. Knowing it's a fern and using the possible species identity provided by Google Lens, I am leaning towards identifying the sample plant as being from the genus Stenochlaena from the family Blechnaceae. The sampled fern does have the Blechnaceae characteristics of sorus arranged in a continuous line near the midrib, fronds reddish in color when young, rachises grooved axially, stipe with more than 2 vascular bundles, and spores monolete. The fern sample also has the Stenochlaena characteristics of moderate to large terrestrial climbing habit, pinnate, dimorphic, rhizome with a complex polycyclic dictyostele and complex multi-stele arrangement in the stipe and rachis, rhizome scales brown, small, usually oppressed, palpate, lateral veins of the pinnae arising from a series of areoles, sometimes close to the costa, a gland, sometimes two, at the base of the lateral pinnae, absence of an indusium, and spore ornamentation. Literature mentions that members of the genus Stenochlaena have been confused with genera Lomagremma, Lomariopsis, and Teratophyllum from the family Lomariopsidaceae. I found that the general frond structure of all these ferns was similar, which perhaps explains why they may be confused. However, the fern genera from the family Lomariopsidaceae can be distinguished from Stenochlaena by venation and other characteristics. In Stenochlaena, lateral veins of the pinnae arise from a series of areoles, sometimes closely parallel to the costa. In Lomagremma, adult fronds pinnae have a closed network of areoles, while in Teratophyllum and Lomariopsis, veins are free with lateral veins of the sterile pinnae arising directly from the costa. Areoles are not present. Reports of Stenochlaena in Malaysia seem to be of Stenochlaena palustris, and the characteristics of the sampled fern do support its identification as being of the genus Stenochlaena and species palustris. Stenochlaena palustris, commonly called Paku Midin or liminding in Malay, is a moderate to large fern identified by rhizome creeping or climbing, yellowish-green to brown, scaly at apex, round in cross-section, 0.5 to 0.7 centimeters in diameter. The rhizome's stele has a radial symmetry with 3 or 4 large bundles surrounded by numerous smaller bundles in 2 approximately concentric circles. Rhizome scales red-brown to blackish, orbicular ovate or ovate lanceolate, margin uneven, palpate often closely oppressed to the rhizome's surface, easily detached. Fronds, 1 pinnate, dimorphic. Stipe, yellowish-green to brown, may possess hairs and scales when young, but is glabrous or almost so at maturity. Grooved on the adaxial face. Laminar ovate or oblong ovate, when young, brownish red, when mature, green, leathery or stiffly papery. Both surfaces glabrous, 4 to 15 pairs of widely spaced lateral pinnae and a larger terminal pinna. Pinnae, except for the terminal pinnae, most articulated to the rachis, sessile to shortly stalked, having a single gland, rarely two, usually present either at the base of the pinnae margin or on the short pinnae stalk, anastomosing veins forming a single row of areoles along each side of the costa. Lateral veins arising from areoles are single or once forked. Sterile pinnae broadly lanceolate, acuminate at apex, cuneate at base, margin sharply toothed and cartilaginous. Fertile pinnae 2 to 3 mm broad, linear, margin entire. Sporangia born in an elongate Cenosaurus occupying the area from the costal areoles almost to the margin of the fertile pinnae. Ex-induciate, paraphysis lacking. Spores bilateral and ornamented with short round projections more or less in rows. Just in case there has been some movement of species from neighboring regions, I decided to see if the fern could be any of the other species of Stenochlaena found nearby. Stenochlaena tenuifolia, unlike the sample, has bipinnate fertile fronds. The sample, like Stenochlaena palustris, has pinnate fertile fronds. Stenochlaena areolaris, unlike the sample, has only 7 pairs of pinnae. The sample, like Stenochlaena palustris, may have more than 7 pairs of lateral pinnae. Stenochlaena milbradii, unlike the sample, has narrow scales on the tip of the rhizome and bases of the pinnae that do not articulate. Conversely, the sample, like Stenochlaena palustris, has orbicular scales at the tip of the rhizome and articulate pinnae bases. Stenochlaena milne, unlike the sample, has sterile pinnae rounded at the base, sometimes articulate, and fertile pinnae are up to 5 mm wide. The sample, like Stenochlaena palustris, has bases of sterile pinnae, cuneate, mostly articulate, and fertile pinnae of only around 3 mm wide. Stenochlaena cumingi, unlike the sample, has sterile pinnae caudate at the base, not articulate, fertile pinnae 7 to 10 mm wide, and spores with continuous or broken ridges. The sample, like Stenochlaena palustris, has bases of sterile pinnae cuneate and articulate, fertile pinnae of only around 3 mm wide, and spores evenly tuberculate throughout. So the sampled fern is indeed Stenochlaena palustris, and I can get the young shoots and leaves and make a delicious sambal to eat. If you want to learn more about this genus of ferns, here is a list of references. If you are interested in fern identification, support the channel with a thumbs up and share the video with others who want to know more about fern identification. Thank you.